So this is an overview of what we'll be covering in today's session. We'll start off with viruses, so structure, life cycle, replication, and its genome. Next, we'll talk about prions and viroids. And finally, we'll talk about bacteria, specifically its structure, gram staining, respiratory obligations, and its life cycle. Starting off with the structure of a virus, not all viruses will have this structure on the right. This is just an example of a bacteriophage. Some viruses will be circular or enveloped, and I'll talk about this in a minute. Viruses are obligate parasites, meaning that they cannot replicate their genomes on their own, so they have to use a host to do so. A virus may contain DNA or RNA to store its genetic information, which can be single or double-stranded. It can be linear or circular, so there's a lot of variation. Viruses do have a capsid, which is a protein coat that is rigid and small. And because this protein coat is small, the virus's genetic information also has to be small. Like I said, some viruses may be enveloped as shown to the right. This envelope is a part of the cell membrane that has been derived from the host by a process called budding. So let's say a virus has entered a cell and replicated inside the cell. The virus's progeny may exit the cell by budding off the membrane. This enveloped virus can then infect other cells by having the envelope fuse with the cell membrane, allowing the virus to enter the cell as a naked genome. Enveloped viruses are especially useful because they're able to camouflage, since they're not able to be recognized by the host's immune system because, like I said, the envelope is derived from the cell. And viruses are also very specific in that they only bind to specific receptors on the cell membrane. So does everyone understand this information about viruses so far? Okay. Because I have a question for you guys. Feel free to put your answer in the chat when you're done. Okay, so the answer is D, like everyone's been saying. Like I said, a virus's genetic information may be written as RNA or DNA. It can be single or double-stranded, and not all viruses will have an envelope. And one thing that is constant with all viruses is a protein coat, which is also called a capsid that surrounds the virus's genome. Does everyone understand? Okay. All right, so let's dive into the virus's life cycle. One phase that viruses may go through is the lytic phase. This process starts off by the virus entering the host and injecting its genome into the cell. The bacterial genome is broken down by hydrolases, which the virus expresses first. The virus will then use the host cell's machinery to replicate itself. And eventually, its genome will be translated into viral proteins used for the virus's structural components. So the virus DNA or RNA will be translated to create complete phages. The progeny will then be released via lysis of the bacterial cell wall, and this is done by lysozymes, which is an enzyme that degrades the cell wall and is also expressed by the virus. The process will repeat itself when the progeny phages infect other surrounding bacteria. The lytic phase is especially advantageous to the virus because it allows viruses to increase in number very rapidly. But it has a disadvantage in that the host cell is destroyed. So evolutionarily, it's not that beneficial. And also, I will be sending you guys the recording and the slides so you don't have to draw this crazy drawing. So because the lytic cycle is not that beneficial, some viruses have evolved to make to enter the lysogenic phase. And this phase starts off with a phage binding to the host cell and injecting its genome. The phage genome will then integrate into the host chromosome. The virus's genetic information will be silenced by the addition of repressors, and the virus's silenced genome will then be referred to as a prophage. The advantage of this phase lies in the fact that every time the host cell replicates, the prophage does as well. So the host cell can either continue replicating or the virus will force the cell into the lytic phase. The lytic phase is usually triggered under stressful conditions. So let's say if the host's survival is compromised, then the virus will want to increase in number very rapidly so that it can replicate and hopefully move on to a new host before its current host dies. So does that make sense about the lysogenic phase? Basically, the whole goal of the lysogenic phase is to create this prophage. And like I said, the prophage is the silenced version of the virus's genome. Okay. So this is the connection between the lytic and the lysogenic phases. So once a virus genome enters the cell, shown by this red star, it can enter the lytic or the lysogenic phase. And like I said, under stressful conditions, it will continue with this lytic cycle. But if the virus feels that it can relax in the host cell, then it'll go through the lysogenic cycle. And here is another question for you guys. Okay, so everyone seems to be on the right track. Does anyone want to explain why the answer is C? Okay, I'll go ahead and explain it. So the question is asking which of the following steps are involved in the lysogenic phase? Like I said, this phase is advantageous because the virus replicates every time the cell replicates. So option A occurs in the lytic cycle. Option B does not occur in the lysogenic cycle. And option D is reversed. So repressors would be added so that the viral DNA would be silenced. And a key feature of the lysogenic phase is that the viral DNA is integrated into the host genome. So before we get into the topic of plus and minus RNA, you should understand the different types of polymerases. As a refresher, a polymerase is an enzyme that creates DNA or RNA from a DNA or RNA template. RNA-dependent RNA polymerase is an enzyme that makes RNA using an RNA template. We understand this by breaking down the name. So the term before dependent is what is used for the template, and the term before polymerase is what the enzyme is making. So RNA dependent means that RNA is a template, and RNA polymerase means that RNA is the product. And here is a practice question. So what does RNA dependent DNA polymerase do? Okay, sorry. So to answer your question, Andrea, the benefit of the lytic cycle is for the virus to increase in number very rapidly. So like I said, um, if a virus is in the lysogenic cycle, but the host is under stressful conditions and it might die, then the virus will trigger the lytic cycle so that it can increase in number very quickly so that it can hopefully infect other bacterial cells before the bacterial cell that it's in right now dies. Does that make sense? Okay. Yeah, everyone is right. So RNA-dependent DNA polymerase uses RNA to make DNA. The viral genome can be of two types, plus RNA or minus RNA. Both are single-stranded and differ in their conformation. So when a plus RNA strand enters a cell, the ribosomes will begin to immediately translate this strand and will make viral proteins. It's important that the plus RNA strand encodes RNA-dependent RNA polymerase. The ribosomes will translate this section of RNA to make this polymerase. And it's crucial that it does this so that the plus RNA strand can be replicated. The host cell doesn't make RNA-dependent RNA polymerase because host cells never make RNA from RNA. And this plus strand can either be translated to make viral proteins or it can be replicated by RNA-dependent RNA polymerase. RNA polymerase will make the complementary strand, which is minus RNA. This minus RNA doesn't do much. It only serves as a template for RNA-dependent RNA polymerase to make more of the plus RNA strands. So if you recall, polymerases make complementary strands. They don't create strands that are identical to the template. So it has to act twice so that the template strand, this plus strand, can be replicated. And finally, all of these plus RNA strands will then be packaged as new viral progeny. Does that make sense for everyone? I know it took me a while to understand this. Yeah, okay. So let's say a virus has a plus RNA genome. So if this phage injects this plus RNA genome into a cell, the plus RNA strand can immediately be translated by the host's ribosome. So it will make viral proteins. And this host ribosome also translates RNA-dependent RNA polymerase from the RNA strand. So once it has translated this enzyme, the RNA polymerase will it will transcribe the plus RNA strand to make the minus RNA strand. And like I said, the minus RNA strand is useless. It doesn't code for any important proteins. So it has to be converted back into plus RNA. You can't go from plus RNA to plus RNA. Like the polymerase always makes the complementary strand, which is minus RNA. So once you have this minus RNA, the polymerase will transcribe it again to make plus RNA strands. Yes, it's always vice versa. Okay. And now we'll talk about minus RNA, which is, like I said, complementary to the plus RNA. Minus RNA, like I said, is useless because it codes for useless proteins. Only plus RNA codes for useful viral proteins. Minus RNA doesn't code for RNA-dependent RNA polymerase, so it has to carry this enzyme inside the capsid. And once the enzyme and the minus RNA strand are inside the cell, the RNA-dependent RNA polymerase, which is already carried in the capsid, it's not translated, will transcribe the minus RNA strand to make a complementary plus RNA strand. As I said before, this plus strand can be translated by ribosomes to make viral proteins, or it can be transcribed by RNA-dependent RNA polymerase to make a bunch more minus RNA strands. And these minus RNA strands will be packaged as new viral progeny. Does that make sense about minus RNA? The main thing to understand is that minus RNA is pretty much useless. It's only used as like a template to make more plus RNA strands. Does that answer your question, Lola? Okay. Yeah, so without minus RNA, we can't make more plus RNA. So a little more on viruses. Retroviruses like HIV have an RNA genome, and these retroviruses integrate into the host genome, but in order to do so, they must go from a single-stranded RNA to a double-stranded DNA. And this is done by RNA-dependent DNA polymerase, which is also called reverse transcriptase. That's pretty much all you need to know about reverse transcription. So before we move on to prions and viroids, does anyone have a question about viruses?
Can you please describe what a retrovirus is again?
Yeah, so a retrovirus is a A retrovirus is a virus that has to integrate into the host's genome, which is usually double-stranded DNA. And in order for this single-stranded RNA to convert into DNA, it needs reverse transcriptase, which is an enzyme, also called RNA-dependent DNA polymerase. And like I said, RNA-dependent uses RNA as a template to make DNA polymerase. Does that answer your question as well, Lola?
Oh, thank you.
Okay, no problem. Okay, so prions are abnormal and infectious proteins. If a normal protein comes into contact with a prion, the normal protein will change conformation such that it is abnormal, similar to the prion. You may be asked about how people become infected with prions. Just know that prions can be ingested or transmitted, inherited, or they can spontaneously arise. To the right is an example of a normal protein and a prion. You can see that the prion has a much different secondary structure. Of course, things can go south when proteins change their conformation because they may no longer be able to perform their essential function in the cell. So individuals can die when infected with prions. One thing that you may have to know or at least recognize is that prions are resistant to heat and proteases because prion aggregates are very stable. That's pretty much all, if not more, that you need to know about prions. Okay, so does anyone have a question about prions before we move on to viroids? Okay. So moving on to viroids, these are different from viruses because they are very simple. A viroid is literally just a short circular single strand of RNA. It doesn't code for proteins and it lacks capsids. The MCAT may just require you to know that viroids infect plants. So it's a very low yield topic. I think I've come across like 1 or 2 questions about viroids when I was practicing. But yeah, it's important to know the difference between viroids and viruses. And like I said, because they don't code for proteins, the only thing they do is silence the plant's genome. And that's pretty much how they cause harm. You don't need to know how they silence the plant's genome, just that they do. So before we move on to bacteria, are there any questions about prions or viroids?
Because I know you said viroids are specific to plants, right?
Yes.
Okay, great. And then for prions, is that like specific to like humans or is it not really specific to anything?
It's not specific to anything.
Okay.
If any, it's animals, I'm pretty sure.
Okay, thank you. I just wanted to make sure.
Yeah. Okay, so prokaryotes are different from eukaryotes because they lack organelles, have a cell wall, and lack a nucleus. A prokaryotic genome is always a one piece of double-stranded circular DNA chromosome. One thing you have to know is that transcription and translation occur simultaneously and in the same place because bacteria lack nuclei. Prokaryotes, I guess as well, eukaryotes have polyribosomes, which is a group of ribosomes that binds to a strand of mRNA. And the goal of this is to translate an mRNA strand simultaneously so it's more efficient. Some bacteria also contain plasmids, and these plasmids might carry antibiotic resistance genes. And we will see in a bit how bacteria exchange information from these plasmids. So the MCAT might give you the name of a bacteria, and they could ask you what the shape of this bacteria is based on the name, or vice versa. So first up, we have round bacteria, which usually are named cocci, or as an example, streptococcus. There's also rod-shaped bacteria that have the name bacilli, for instance, Bacillus anthracis. Finally, spiral bacteria are usually called spirochetes. An example would be spirillum. Yeah, spirillum minus. But the names of spiral bacteria could vary. The way I thought of it was if coccus or bacilli wasn't in the name, then it's spiral. The way I distinguish between round and rod-shaped bacteria is that coccus has a lot of C's in it, and C's are round, so round bacteria are cocci. Not sure if this will work for you, but I don't know. Anything helps me, so. Another high-yield topic is membrane staining. The bacterial cell wall is made up of peptidoglycan, which is just a polymer that retains a purple stain. There are generally two structures that a bacteria cell wall could have. It can be gram-positive or gram-negative. So gram-negative bacteria have two layers of cell membrane. The outer layer consists of lipopolysaccharide, which are referred to as endotoxins and can trigger an immune response. Gram-positive bacteria only have an inner layer of cell membrane. The outer layer consists of a thick layer of peptidoglycan. And notice that gram-positive bacteria do not have lipopolysaccharide because they lack this outer membrane. You don't need to know the exact details of staining, but more so the results of it. So just how it works in 4 simple steps is you apply a violet dye to a group of bacteria. Then you apply iodine, followed by washing with alcohol to bring about decolorization, and finally applying a counter stain to the bacteria that are not stained purple. Again, you don't need to know this. I just wanted to give you some background information. Gram-negative bacteria will stain pink because the outer cell membrane is covering this layer of peptidoglycan. So it's not accessible and therefore will stain weakly. Gram-positive bacteria will stain strongly and will appear violet because the peptidoglycan is able to be stained and retain this violet dye. The way I remembered these two results is that gram-negative is negative for purple, so it will stain weakly, or pink. Whereas gram-positive is positive for purple, so it will stain purple. And why will it stain purple? Because it has that thick outer layer of peptidoglycan, which retains purple dye. And if anyone has an easier way of remembering this distinction, please type it in the chat. But does anyone have any questions so far? Okay. So the flagella is an integral part of a bacterium because it allows for motility. There are three different arrangements of flagella. You don't have to know the names, but sometimes passages will give you so many details. So it at least helps to recognize these details instead of getting worried about not knowing this term and wondering if a question has anything to do with it. Okay, so the three types are monotrichous, which means a bacterium has one tail. The second one is amphitrichous, which means two tails at both ends, and peritrichous, which means multiple flagella. Bacteria do have a flagella hook and basal structure. Again, you don't need to know too many details about this. You do have to know that it is different from the eukaryotic flagella because it does not have a 9+2 microtubule arrangement. And what I mean by 9+2 microtubule arrangement is just the arrangement of microtubules around the hook. So basically the microtubules that are arranged in a specific manner to create this flagella. Does everyone understand? All right. So when you're done answering this question, please type it in the chat. Okay. Okay, so everyone seems to be on the right track. The answer is A, and you are looking for a bacteria that's rod-shaped. C and D have the word coccus or cocci in them, and these, like I said, are round. B has the word spirillum in it, and that sounds like the bacteria's spiral, so A is our answer. And yes, I'll go to the previous slide. Let me know when you have the information that you need. Is it okay if I move forward, or is there something that I could explain? Okay. And I have another question for you guys. Okay, everyone is on the right track, and this makes me very happy. So does anyone want to explain their answer?
When I read that it was pink, I immediately eliminated the positives because those ones are dark purple, and then I saw that it was round, and then I liked your memorization technique. You said like C is kind of round, and so I just picked the one that was coccus negative.
Yeah, great. That's awesome. I'm glad my weird technique helped you. Okay, so we can move on. And now we'll get into classifying our bacteria in terms of survival conditions. We'll start off with temperature. Psychrophiles love to be in extremely cold temperatures, so about 0 degrees Celsius. Mesophiles are kind of like us, they're in the middle, so they'll live in moderate temperatures of about 30 degrees Celsius. Thermophiles love heat, so they'll be found in temperatures of about 100 degrees Celsius. This one's easy to remember because you have the word thermo in there, and I guess also meso means middle, so yeah. And we can also classify bacteria according to their nutritional requirements. Beginning with autotrophs, these bacteria will use CO2 as a carbon source to build their carbon backbone. Heterotrophs are kind of like us. They will use organic nutrients created by other organisms as a carbon source to make their own organic molecules. And in terms of how bacteria obtain energy, chemotrophs will get their energy from chemicals, so chemochemicals. Yeah. And phototrophs will get their energy from the light, so like plants. Bacteria can be chemoautotrophs, chemoheterotrophs, photoautotrophs, or photoheterotrophs. Chemoautotrophs will build carbon molecules from CO2 and will use energy obtained from chemicals. So you get that auto aspect that the bacteria is able to use CO2 to make their own molecules, and the chemo aspect that in order to do so, they need energy from chemicals. Chemoheterotrophs get their energy from the same place as chemoautotrophs, from chemicals. But this type of bacteria cannot use CO2 to make their own carbon molecules. Like I said, they're part heterotrophs, so that means they're like us. They must obtain organic molecules from other organisms because, like I said, they're part heterotroph. Photoautotrophs, because they're part autotroph, can use CO2 as a carbon source, and because they're part phototroph, obtain energy from the sun. Photoheterotrophs also obtain energy from the sun, except they're not able to use CO2 as a carbon source. So they must use organic molecules from other organisms. The key to distinguishing these is to break the word down into their components. So like phototroph, autotroph. Does that make sense? Okay. Auxotrophs are mutants that require an additional nutrient that an otherwise wild-type strain of bacteria can synthesize on their own. So the way these auxotrophs are written is that an amino acid will have a superscript of a minus sign. This means that the auxotroph is not able to synthesize that amino acid. But if it has a plus sign, it means it can synthesize this amino acid. So let's say Arg minus means that the auxotroph can't make arginine, and it must be given arginine for it to grow. This nomenclature is a bit different for enzymes. So lac minus means that the bacteria cannot metabolize lactose. So if it's given lactose, then it won't survive. Does the nomenclature make sense? Okay. Here's a question. Okay, so the correct answer is B, but does anyone want to explain why?
I
Okay.
looked at that. It has it obtains energy from chemicals, and so I looked at chemo, so that took out C and D as options. And then since it's able to incorporate carbon dioxide into sugars on its own, the on its own part means like independent, so autotroph.
Yes, that's perfect. Thank you so much for telling us. Okay. No, I'm sorry. Okay, so as we know, bacteria can be classified according to their tolerance for oxygen as well. So bacteria that require oxygen for survival are obligate aerobes. They are obligated to be in an environment with oxygen. Facultative, I don't know if I pronounced it right. Facultative anaerobes will use oxygen if it's available, but they don't need it to survive. Tolerant anaerobes simply tolerate oxygen. So if oxygen is present, they'll just ignore it, they won't use it, and they'll continue surviving. Obligate anaerobes, on the other hand, neither use nor tolerate oxygen. So if oxygen is present, they'll die. As we know, bacteria reproduce asexually. And let's say you culture bacteria in a media with nutrients. This initial phase will be the lag phase. And during this time, bacteria will not grow even though there are nutrients, because the bacteria are synthesizing the necessary enzymes and proteins needed for replication. Once it has all of this machinery prepared, the bacteria will begin dividing exponentially. So that's where we will see this log phase. Eventually, the population will get so big that the plate runs out of nutrients. So the bacteria stop growing and they just stay stationary. Once enough time passes and the bacteria starve, they'll start to die because no additional nutrients are being provided and the medium can no longer support their growth or survival. So that's why you will get this dead phase. Does anyone have any questions about the growth and life cycle? Okay. Here's a question for everyone. Yes, the correct answer is B, because the bacteria is able to use oxygen, but it doesn't require it for its survival. So it's a facultative anaerobe. A is wrong because the bacteria will survive in the presence of oxygen. It just doesn't use it. A and C are wrong because the bacteria doesn't require oxygen to survive. There are 3 ways that bacteria can transfer genetic information to each other. The first one is transduction. Transduction involves the use of a bacteriophage that transfers a section of DNA from bacteria A to bacteria B. If we remember from the lysogenic phase, repressors are added to silence the viral genome. But when the virus is ready to enter the lytic phase, so it can create progeny that will infect more bacteria, this repressor has to be taken out so that the viral genome in red can exit outside of the host chromosome. The enzyme will not make this cut perfectly, so when it tries to excise the viral genome, it will take out a bit of the host chromosome, and eventually the viral genome now consists of a small section of the host's genetic information. I tried drawing this on the right. So in the bacterium on the bottom, just before it's about to enter the lytic phase, the viral genome has to be excised, like I said. So let's say the enzyme cuts at the red lines, then the viral genome will look like this circle on the right after the red arrow that includes this black portion, which represents the host chromosome. When these progeny go on to infect other bacteria, it will introduce information from the little piece of the host chromosome to the new bacteria. Does everyone understand how that information is transferred? Okay. The second method for genetic exchange is conjugation, which is the process whereby two bacteria create a bridge. The male cell, also known as the F positive cell, will connect its sex pilus to the female cell, or the F minus cell. And these two cells will become connected such that the F plasmid will replicate and transfer to the F minus cell, and eventually the F minus cell will become an F positive cell. What do I mean by F? I mean the fertility factor, which is an extra chromosomal element that's referred to as the F plasmid. This plasmid may carry information for antibiotic resistance, for example. It's just important to know that the F plus donor will replicate its F plasmid and transfer that to the F minus cell. And the end result is two F positive bacteria. So for example, before conjugation, the F minus cell lacked this F plasmid, which carries a penicillin resistance gene. Once conjugation occurs, the F minus recipient will now be able to survive in penicillin and will also be an F positive bacteria because it has this F plasmid. Does conjugation make sense for everyone? Okay. This is where things get slightly complicated. You don't need to know all the details. I'll just tell you what the MCAT would want you to know. So HFR stands for high frequency of recombination, which means that the bacterial chromosome undergoes recombination with the F plasmid, so the green and purple. If you don't recall what recombination means, it just means crossing over of genes and chromosomes. So it's a way to exchange genetic information. The F factor in an F positive cell will integrate into the host chromosome in green. When F minus female bacteria comes along, the F positive cell will make this bridge. Similar to regular conjugation, the F factor will be replicated and transferred to the F minus recipient, aka the girl. In order for it to be transferred, it has to be excised from the bacterial chromosome. Like I said, not all excisions are perfect, so it will actually take some of this bacterial chromosome with it. The end result is that the F minus recipient will have this F plasmid that also consists of some of the donor chromosome. And because this F plasmid has a high frequency of recombination, it will undergo recombination with the recipient chromosome. You can see that the genes in the recipient chromosome are lowercase letters, and the donor genes on the F plasmid here are in capital letters. So let's say A minus stands for not carrying the enzyme required to synthesize arginine. The A plus gene on the F plasmid does carry this information, so it will recombine with the host chromosome. And now the recipient chromosome can code for this enzyme. This process is useful for mapping, so scientists will often stop conjugation in small intervals to see which genes are transferred first. So A would be transferred before B. Allowing conjugation to continue for a longer period of time will allow for more of the donor chromosomal genes to be replicated and transferred. So if the bacteria starts displaying the phenotype of A before B, then we know that the gene A is earlier than gene B. Does that make sense? And this was a lot for me to understand at first. Okay. Because I have a question for you guys. Okay, so the correct answer is B. The question is doing what I said previously. The scientist is stopping conjugation at short intervals so they can map out the genes. The recipient bacterium expresses the gene for the enzyme to make leucine first, followed by histidine, and then followed by arginine. So we can say that the gene for leucine occurs first, then histidine, and then arginine. Does that make sense? Because, like I said, the bacteria is starting to display the phenotype of leucine first. So Leu plus. Okay, so after 3 minutes of conjugation, the recipient bacterium is Leu plus, meaning that it's able to make leucine. So the scientist is noticing that the bacteria is able to make leucine. So they conclude that the gene that codes for the enzyme to make leucine occurs first. Then the scientist allows conjugation to continue, and they notice that the bacteria is able to make histidine now. So they're able to conclude that the gene that codes for the enzyme to make histidine occurs after leucine. But the scientist is noticing that the bacteria cannot make arginine yet. So conjugation is allowed to continue again. And now the scientist notices that the bacteria is able to make arginine. So the gene that codes for the enzyme to make arginine is after histidine. Does that make more sense? If at the end it was still negative, would it even be counted? In that case, conjugation was probably not allowed to continue for a long enough time, or simply that the enzyme cut the chromosome before that gene was, I guess, integrated into the plasmid. So remember how I said that enzymes will excise the viral genome, the F plasmid imperfectly, so it will include some of the chromosome with it. If at the end it was still negative, then the gene was probably left out. This is a visual representation of this bacteria. So like I said, the scientists stopped conjugation at this squiggly line, and that's how they noticed that the bacteria was able to synthesize leucine. Then conjugation is allowed to continue. They stop it again at the squiggly line. So they noticed that the bacteria was able to make histidine, so they predict that the gene for histidine lies here. Then conjugation continues again, and this time the bacteria makes arginine, so they can say that the gene for arginine is after the gene for histidine. Does that make more sense, hopefully? Okay. And last but not least, we have transformation. This process just involves the bacteria taking in naked DNA strands from the environment, and it's a way for bacteria to obtain genetic information from other bacteria. I'm not sure how bacteria just release these free DNA strands. The MCAT doesn't require you to know the details, just what this process is and to differentiate it between conjugation and transduction. So that's actually all the content I have for you guys today. Do you want me to clarify anything? I know that the HFR thing was pretty complicated. Okay. So thank you for your patience and staying for the full session. If you could please donate to our PayPal so we could keep these sessions going, that would be very much appreciated. Each donation comes with a cool holographic sticker. And you can also join our Facebook group. This is useful for finding study buddies and making friends, possibly meeting people who are in the same stage as you. And you can also email us at mcat101questions@gmail.com. So look out for the post-topic session email that will be sent out later tonight, and it will also come with the recording and a copy of the slides. When do we study this biochem? This is under general bio. Alola, what do you mean by practice question? No problem. Thank you for coming. Thank you for your thank yous. Would there be another session where we could? Yeah, so I will plan that out. I'll talk to Sim and see if I could schedule one next week. I'm also trying to make a session where we go over practice passages, and hopefully the next one will focus on microbiology, especially since those are pretty high yield. So yeah, keep an eye out for that. All right, thank you everyone for your thank yous. I hope you have a good night and good luck studying.